Okay. Good morning, everybody. I'd like to. Hello, everybody. Good morning. I'd like to start our poster discussion session. And for the first poster presentation, I invite Umut Ulas Yeslurt with the presentation Characterization of Adhesion and Migratory abilities of epithelial and mesenchymal pancreatic cancer cell populations with different cancer stem cell properties. Please.
First of all, I would like to thank you for the opportunity to present my project at the EPC. My name is Umut Yeşilyurt and I'm a medical student from Germany. The aim of my doctoral thesis was to characterize the adhesion and migration abilities of pancreatic epithelial and mesenchymal tumor cells with different stem cell properties. Pancreatic ductal adenocarcinoma is characterized by its early metastatic potential, which often limits the treatment of patients. To better understand the mechanisms of metastasis, in particular the migration of tumor cells and their adhesion to endothelial cells in the periphery, I analyzed the in vitro potential of two different PDAC cell lines and their subclones. The cell lines were generated in a single cell dilution assay and later characterized with respect to their EMT and CSC phenotype. As you can see, the PANC1 cells show a more mesenchymal phenotype, while the PANC89 cells are more epithelial. For the adhesion, I first seeded lung endothelial cells on the bottom of the wells, and after 24 hours, I placed the tumor cells on top. Before the measurement, I washed the cells with PBS to remove non-adherent cells, and the migration behavior was analyzed with a scratch assay using EBD inserts to obtain standardized gaps between the two cell fronts. Now to the results of my experiments. There were no differences in adhesion to lung endothelium between the subclones of the two cell lines, as you can see on the graph on the left. In contrast, the migration potential of epithelial PANC89 cells with CSC phenotype was significantly higher than that of the hybrid non-CSC PANC89 cells, as you can see on the graph on the right. The PANC1 cells showed no difference in migration. In fact, they showed no migration at all. The comparison between the PANC1 and PANC89 cell lines showed again significantly higher migration behavior of the PANC89 cells compared to the PANC1 cells. The immunohistochemistry and immunofluorescence staining showed that the PANC89 cells migrate mainly as cell clusters, as you can see on the images, and that these clusters consist of epithelial CSC cells and non-epithelial non-CSC cells. Overall, my results show that there's no difference in adhesion to the lung endothelium of the different PDAC cell clones. In contrast, the epithelial PANC89 cell lines migrate significantly faster than the mesenchymal PANC1 cell lines. Furthermore, PANC89 subclones with a CSC phenotype migrate faster than PANC89 cell clones with a non-CSC phenotype. The stainings show that PANC89 cells move primarily as clusters rather than as single cells. These clusters show expression of both epithelial and stem cell markers, but also of neither. Therefore, it can be said that migration, in contrast to adhesion, is dependent on both the EMT and the CSC phenotype, and that the association of the cancer stemness and epithelial to mesenchymal transition has a positive impact on PDAC metastasis. Thank you for listening.
Okay, thank you for the interesting presentation. Have you any questions from audience? No? Okay. I'd like to clarify how and where you received pancreatic epithelium and mesenchymal cancer cell population.
As I have already told, we in our lab we made a single dilution assay of two cell lines that were generated before, so the PANC1 and PANC89 cell lines. And in the single dilution assay, we characterized the cells regarding their ability to form colonies. And these colonies were later characterized with stem cell and epithelial markers. So the stem cell markers were SOX2 and nestin, and the epithelial marker was ecadherin. And after characterization via PCR, we have decided that the cells with a high epithelial expression of E-cadherin are epithelial ones, and the cells with no E-cath expression are more mesenchymal-like, and that cells with a high expression of ZOX2 and or nestin are stem cell cells, or cells with a more stem cell-like phenotype.
Thank you very much. And okay, the next report will be made by Amelie Modi. I invite you with presentation impact of cancer stem cell markers nestin and SOX2 on cell migration of epithelial and mesenchymal pancreatic cancer cells.
Hello, my name is Amelie Modi and I'm a medical student. I'm currently working on my doctoral thesis at the Institute for Experimental Cancer Research at the University Hospital Schleswig-Holstein in Kiel, Germany. And today I would like to present my poster on the theme impact of cancer stem cell markers, nestin and SOX2 on cell migration of epithelial and mesenchymal pancreatic cancer cells. So, first of all, the pancreatic ductal adenocarcinoma, short PDAC, is one of the most frequent cancer-related causes of death. Cancer stem cells, short CSC, contribute to tumor progression and metastasis, and thereby to poor prognosis. So the aim of the study was to investigate whether and how knockdown of the stem cell markers nestin and SOX2 affects migratory and cell growing abilities of parental mesenchymal, PANC1, and epithelial PANC89 PDAC cells and their isolated CSC clones. For this matter, a migration assay was conducted. Knockdown of the stem cell markers were performed by SE-RNA transfection, and the cell growth was determined by cell counting. The cell migration was analyzed using 2 cavity inserts, by which standardized gaps between 2 cell surfaces were created, and then the gap closing was measured over time. On the right side, you can see the successfully suppression of nestin and SOX2 expression. And now I like to move on to my results regarding cell growth and migration. On the left-hand side, you can see that the cell lines and their CSC subclones differ in their cell growing behavior. And more importantly, we could observe also in their migratory abilities. Despite the epithelial phenotype, the untreated PANC89 cells and especially their CSC subclone showed pronounced cell migration here in green. In contrast, the untreated mesenchymal PANC1 cells and their CSC subclone showed little migration, here in purple. As you can see here in the top, cell growth of both epithelial and mesenchymal cells was decreased after knockdown of both stem cell markers. The cell migration of the epithelial cell line PANC89 seemed to increase under nestin knockdown, particularly those of the cancer stem cell subclone, here in light green, and the nestin knockdown did not impact the migratory abilities of the mesenchymal cell line. The SOX2 knockdown seemed not to influence migratory behavior of both cell lines and their CSC subclones. To quickly summarize this, the data indicate that nestin and SOX2 both promote cell growth of both PDAC cell lines, and only nestin seemed to inhibit migratory abilities of epithelial PDAC cells, particularly those with the CSC phenotype. Overall, these findings could contribute to a better understanding of cell migration and metastasis of the PDAC. Thanks for your attention.
Okay, we have now a question from the audience.
May I ask a question?
Oh, yes, Professor Stahl.
Nice talk, nice work. Do you think that this phenomenon of nesting is present in any cancer, pancreatic cancer, or maybe it may be different in different subgroups, let's say, based on let's say classical or subtype, let's say?
I'm so sorry. Would you repeat your question? I'm sorry.
Yeah, sorry. Do you think the phenomenon of nesting, which is expressed in many cancers also, do you think it is common to any pancreatic cancer, or maybe there may be differences according to the different cell line or different subgroup of pancreatic cancer?
I cannot say for sure, but I could imagine that it would be maybe different, because in also my in the cell lines there were different levels of nestin, and so I could imagine that the impact would be different.
Okay, thank you very much.
Sure.
Okay, thank you very much, Emily. And the next presentation, for the next presentation, I invite Philip Hartnett with presentation impact of targeted CHP2 inhibition, MPA-PK inhibition on metabolic rewiring in pancreatic cancer.
Hello and welcome to this poster presentation with the topic impact of targeted SHP2 inhibition plus minus MEK inhibition on metabolic rewiring in mutant KRAS-driven tumors. So recent work established a profound inhibition of tumor development by deletion of PDPN11, encoding the non-receptor phosphatase SHP2 via reduction of oncogenic KRAS activity. Our group leader, Dietrich Ruiz, described a strongly synergistic treatment of SHP2 inhibition in combination with MEK inhibition to treat KRAS-mutated pancreatic cancer. My aim is to investigate the influence of genetic loss or pharmacological inhibition of SHP2 plus minus MEK inhibition on the metabolism of KRAS-mutated PDAC cells. So in figure 1, we started to investigate how the pharmacological inhibition of SHP2 by the SHP2 inhibitor SHP99 or the genetic knockout of SHP2 influences the proliferative behavior. By adding metabolic inhibitors, we observed an increased sensitivity displayed especially by the SHP2 knockout cells for oxidative phosphorylation and autophagy inhibitors. In figure 2, you see supposedly resistant cells, and that the adding of SHP2 inhibitor SHP99 possibly breaks the resistance here. The additional adding of the metabolic inhibitors display a possible interesting effect. So the YAPC cells are able to proliferate in presence of the autophagy inhibitor chloroquine, which indicate that these cells are not that dependent on autophagy in comparison to the unresistant cells seen in figure 1. But for sure, these data are very preliminary and we have to investigate this in detail. In figure 3, we investigated the autophagic behavior by using an approach to stain the autophagosomes. In PANC1 cells, we observed a higher level of basal autophagy in SHP2 knockout cells. In addition, our results support the findings observed from Jenning Dare et al. That the initiation of the MAPK pathway leads to an increase of autophagy. In addition, the SHP2 knockout cells treated with trametinib indicate an increased autophagy in comparison to the knockout cells treated with DMSO. Since the proliferation assay from figure 1 indicate a higher dependency of SHP2 knockout cells for oxidative phosphorylation, we checked the respiratory capacity by using the Seahorse device in figure 4. So we observed no significant differences in basal respiration, but clear difference in respiratory capacity. In figure 5, you see RNA sequence data from PANC1 cells. Treating the cells with trametinib results in 179 altered expressed gene sets in comparison to DMSO-treated cells. In the combinational treatment, we observed over 600 altered gene sets, which confirms the synergistic effect of this treatment. And it is noticeable that in relation to others, we have more affected metabolic gene sets in comparison to the trametinib single-treated cells. And we see a way more metabolite pathways that were detected. In conclusion, this work in progress data indicate that the dual SHP2 MEK inhibition seem to induce considerable changes in PDAC metabolism, which might be therapeutically exploitable. Thank you very much for your attention.
Yeah. Thank you very much, Philip, for very interesting presentation. Have you any questions?
Yes, I would like to ask a question. Do you think that the type of KRAS mutation makes a difference?
Yeah, that's a really good point. So currently we are testing different cell lines with different KRAS mutations. And yeah, for sure, there could be some differences, but so far we have no clear indications or something in that direction.
Okay, so thank you very much.
No. Thank you very much for interesting presentation. And for the next poster presentation, I invite Eleonora Gall, the effect of bile on pancreatic cancer, the importance of mucins.
My name is Eleonora Gall. Today, I'm going to talk about the effect of bile on pancreatic cancer and the importance of mucins. Pancreatic cancer is one of the leading causes of mortality rate globally and usually associated with obstructive jaundice. Our aim were to characterize the effect of bile acid on carcinogenic processes and to investigate the underlying mechanism. In the first step, we measured the serum level of bile acids in pancreatic ductal adenocarcinoma patients with or without obstructive jaundice and in healthy volunteers. We found the highest level of bile acids in the pancreatic ductal adenocarcinoma patients with obstructive jaundice. Then we treated kappa-1 cells with this human serum and we found changes in the cell morphology. So we vimentin immunohistochemistry. Vimentin is a protein which expresses in the mesenchymal cells, but not in the epithelial cells. We found a very strong vimentin staining in the PDC-OJ serum treated group. Then we wanted to know the effects of bile acid on cancer progression. So we treated kappa-1 cells with 6 different bile acids in 2 different concentrations for 24, 48, and 72 hours. We found most of the bile acids time and dose dependently increase the rate of proliferation, adhesion, migration, invasion, and the ability of colony forming of the cancer cells. And we also investigated MAC4, which is a mucoprotein, which is a biomarker in pancreatic cancer also, and overexpression of this isoform is usually associated with poor prognosis. In kappa-1 cells, after bile acid treatment, we found most of the bile acids dose and time dependently increase the expression of this isoform, both in mRNA and in protein levels. In human samples, in normal pancreas, and in the case of neuroendocrine tumor, we found a totally negative staining, but in the case of pancreatic ductal adenocarcinoma with or without obstructive jaundice, we found a very strong MAC4 staining. The 4-year overall survival rate was significantly poorer in the PDC-OJ group compared to the PDC group. MAC4 silencing, we found MAC4 silencing significantly decrease the carcinogenic effects of bile acid in kappa-1 cells. So taken together, we could say in pancreatic cancer patients where the disease is associated with obstructive jaundice, the early treatment of biliary obstruction can make life expectancy. And thank you for your attention.
Thank you very much, Eleonora. We have one question from the chat. For whom of others? I don't know.
I can see that. Maybe Claudia Merlentin is asking, thank you for your interesting presentation. It is known that gallstones are a risk factor for pancreatic cancer. Do you have any idea what the mechanism of this could be? Eleonora, this is a question to you. Eleonora, are you online? maybe that there is no connection. Okay, so if we have no answer, maybe we can go ahead with the next presentation by Ren Lei on neuronal glutamate.
Hello, hello, I'm Lei from Professor Academia. My postdoc is about pancreatic cancer and urines. As very low pancreatic cancer show a very stronger propensity to renal invasion, but its functional mechanism is not clear. So here we have set this glutamate AMD signaling pathway at the synapse level, which could be also involved in the interaction between pancreatic cancer and urines during renal invasion. As well, glutamate is the ligand of AMD, many present in synaptic vesicle, which could be trafficked into presynaptic membranes for the actual synthesis by the vascular glutamate transporter 2 proteins. Cerebral brain 1 is an essential membrane component of the synaptic vesicle, which could also involve in the actual synthesis by bringing the target membrane and the synaptic vesicle to be more closely and allow them to fuse. And then glutamate will release to the synaptic cleft and cross the cleft and bind to the binding site of glucan 2 subunit with the help of PSD195, which could anchor and stabilize the surface expression of glucan 2 containing MDR and regulate the interaction between MDR and the downstream signaling molecules. So here we first profile the glutamate receptor profile of human pancreatic cancer on TCGA and TCLE dataset, and we contact this pattern to the neuroimmature pancreatic cancer cells and the non-neuroimmature pancreatic cancer cells, and then we check their level by qPCR. Finally, we focus on GLUT2D gene, and then we performed immunohistochemistry to check and we found that the transduction molecules of this glutamate AMD signaling were activity in human pancreatic cancer tissues with neural invasion, and which could also be activated by DRG condition medium and work culture with DRG. This result we didn't show here because of the limited area. And then in the co-cultures of urines and pancreatic cancer, we found that glucan 2D, glucan 1, and the PSD was significantly upregulated in neuroimmature pancreatic cancer cells, and also we found the same cerebral brain 1 and glucan 2 was significant on the actions extended from neurons and on the cancer cells, but we didn't observe this on the non-neuroimmature pancreatic cancer cells. And from the functional mechanism, glutamate and DRG condition medium treatment led to EZH2 enrichment and glucan 2D promoted by EZH2 ETF1 signaling pathway. So finally, we conclude that neurons promote the migration of pancreatic cancer towards to neurons in glutamate fuel way by glutamate AMD signaling, which is regulated by the EZH2 ETF1 upstream regulatory signaling pathway. Okay, thank you. That's all.
Thank you very much. May I ask you if do you think that there is any way to block this phenomenon?
Sorry, can you repeat your question? Sorry.
Yes. Do you think there is any way to block this phenomenon in cell lines at least, or in your experimental model, so that we can imagine we can block this in patients?
Yes. First we pick up some female blocks, tissue blocks, and check this transduction molecules by the immunohistochemistry, and based on before this we perform this pattern in the neuroimmature pancreatic cancer cell lines and non-neuroimmature pancreatic cancer cells. Because of the space is not enough, so we didn't show our result here. Based on the finding, based we found the result on the CNS, and then perform the result. We perform the experiment on human tissues. So yeah. So we will write do all of this.
Thank you very much. Very interesting. So do you think we can move to the next presentation by Maria Blauer on the organization of pancreatic acinar cells regulated by stellate cells?
We thank the organizers for the opportunity to present our work in this meeting. The title of this presentation is Organization of Pancreatic acinar cells is regulated by stellate cells in a three-dimensional co-culture model. My name is Merja Bloyer, and I work as a researcher with the Tampere Pancreas Group, Finland. We have nothing to disclose. Together with ductal cells, pancreatic acinar cells and stellate cells are central players in exocrine pancreatic pathobiology. Culture techniques facilitating their interactions in a three-dimensional setting provide valuable experimental tools for pancreatic research. In this work, a three-dimensional culture model was optimized to support acinar-stellar interactions in vitro. Here you can see a schematic illustration of the sandwich culture setting. We used here normal mouse acinar and stellate cells that were obtained using the explant outgrowth technique. 20,000 cells were seeded between two matricial layers. Both acinar and stellate cell monocultures and co-cultures were maintained for 4 to 5 days in serum-free medium, and their development was monitored under a phase contrast microscope. In control co-cultures, fibroblasts were substituted for stellate cells. In monocultures, acinar cells organized into hollow spheroids. Spheroid fusion started from the first day of culture, and continuous expansion in spheroid volume was observed. Stellate cells in monocultures grouped together into dense clusters. In acinar-stellar co-cultures, epithelial buds emerged from clusters of stellate cells. In control co-cultures with fibroblasts, however, acinar cells formed hollow spheroids. In conclusion, a three-dimensional culture model was developed and optimized to support acinar-stellar interactions in vitro. The presence of stellate cells, but not fibroblasts, facilitated acinar cell organization into bud-like structures resembling those seen during branching morphogenesis. Acinar-stellar interactions prevented uncontrolled growth and fusion of acinar cell spheroids, typical of acinar cell monocultures. Thank you for your attention.
I ask, do you think that this phenomenon is also important in carcinogenesis, this interaction? Because now that there is the feeling, some data, that there are acinar cells that may become cancer and ductal cells that may become cancer. They give origin to different types of ductal adenocarcinoma, pancreatic carcinomas. Do you think there is the importance of these cells in this phenomenon?
Yes, thank you for your question. In this work, we used normal acinar cells.
Yeah, sure.
That we have previously shown to produce amylase, for example. And I don't know, I have the feeling that this may be a sort of healing process, and I'm not sure if this could be related to cancer, but anyway to some sort of injury. This is my idea.
Yeah, well, you know, I mean, cancer is thought since many years that is a healing process that is uncontrolled. So maybe this is a nice answer. Thank you very much.
Thank you.
Thank you. So we can move to the next speaker, who is Malte Buchholz, who will talk us about microRNAs and POR3K.
My name is Malte Buchholz, and I'm giving this talk on behalf of Yajing Yang, who is in the process of relocating to the US at the moment. POR3K is a subunit of RNA polymerase III, which is responsible for transcription of tRNA and other small non-coding RNAs. And we had discovered it to be potentially overexpressed in pancreas cancer, which we also confirmed via real-time PCR in a small sample size here of tissues from cancer, chronic pancreatitis, and normal tissue. So we followed this up by siRNA experiments using 3 independent siRNAs and 3 pancreas cancer cell lines. And we saw profoundly reduced cell viability upon knockdown, which went along with an upregulation of P21 and a downregulation of cyclin D1. And we saw increased necrosis, but no signs of induction of apoptosis in these cells. And the picture was even stronger under 3D cell culture conditions. Here, the viability was much stronger reduced in the knockdown conditions, while cell migration, for example, was not affected. So we reasoned that maybe microRNAs would be mediators of this effect of POLR3K, and we found that MER30D is specifically downregulated upon the knockdown of POLR3K. So we decided to do rescue experiments using RNA mimics for MER30D, and as you can see, these mimics did not affect the expression of POLR3K itself, nor the knockdown efficiency. If you compare the control-treated cells with cells that received an siRNA plus a RNA mimic control, there's still the growth inhibition, but this could be effectively rescued using a MER30D mimic. Going back to primary pancreas tissues, we used the tissue microarray to stain for POLR3K, and you can see in normal tissues there's a staining of the cytoplasm, which changes more towards the nucleus in the tumors. But neither the overall expression levels nor the specific localization in the cell was directly correlated with patient survival. So what I hope I was able to convince you is that POLR3K is overexpressed in at least a subset of human pancreatic cancer. It controls cell growth, especially under 3D conditions. It affects anchorage-independent growth, in part at least through MER30D, and the subcellular localization varies, but is not directly correlated to patient survival. With this, I thank you for your attention, and I'm happy to take any questions.
Yeah, thank you. Which subtype? In your conclusion, you said that the expression and so the phenomenon is in the subtype of pancreatic cancer. You know which subtype?
No, I said it's in a subset of cancer, so it's not overexpressed in every cancer. We haven't actually checked a correlation with the subtypes, the known subtypes, but I would not expect a correlation with subtype because we did not see a correlation with survival either. And we know that that the subtypes differ in survival, right? So if there was a strong correlation with a specific subtype, we would expect to see a difference in survival correlated with POLR3K expression, but we don't see that.
Okay, thank you very much. So are we moving to the next speaker, who is the Noemi Salzani-Papp on the inhibition of mitochondrial sodium-calcium exchanger, preventing acinar cell damage in acute pancreatitis in mice.
Dear audience, my name is Noemi Salzani-Papp, and thank you for the opportunity to present my poster. Unfortunately, due to lack of time, I can only present part of the poster now, but all the details can be found on my poster. Mitochondrial calcium homeostasis is essential for regulating cellular bioenergetics, calcium signaling, and cell fate. Under physiological conditions, mitochondria accumulate calcium via the mitochondrial calcium uniporter, MCU, whereas calcium is extruded by the mitochondrial natrium-calcium exchanger, NCLX. The balance between these processes is essential for preventing sustained mitochondrial calcium overload, which is hallmark of acute pancreatitis. Based on previous experiments, NCLX may play a critical role in the development of mitochondrial calcium overload. The aim was to clarify the role of NCLX in pancreatic acinar cell during acute pancreatitis. NCLX and MCU are expressed in mouse brain, pancreatic tissue, pancreatic acinar cells, and ductal organoids as well. MCU and NCLX expression show significant difference in the cellular treated group compared to the control treated group. and we see the immunofluorescence labeling of NCLX in isolated acinar cells. 250 micromole cannadesoxycholic acid, CDC, treatment induced a sustained mitochondrial calcium elevation. The inhibition of NCLX by 10 micromole CGP moderately restored the mitochondrial calcium efflux of the pancreatic acinar cells. Average traces of mitochondrial membrane potential is response to CDC show a significant decrease in the mitochondrial membrane potential. Average traces of mitochondrial membrane potential is response to CDC and CGP together show a significant lower drop of the mitochondrial membrane potential compared to the CDC treated group. Experimental acute pancreatitis was induced by 10 intraperitoneal serum injection or 2 intraperitoneal injection of ethanol and palmitic oil acid together. In all cases, we were able to demonstrate the protective effect of CGP in the basis of histological parameters and in serum amylase levels. Our results suggest that NCLX plays a crucial role in the mitochondrial calcium homeostasis in pancreatic acinar cells under pathophysiological conditions and can be a potential drug target in acute pancreatitis. Thank you for your attention, and thanks my supervisors and the entire research team for their help.
Okay, thank you very much, Naomi. Tell me please, are there currently any agents in the clinic that can inhibit the sodium-calcium exchanger? Tell me please.
Sorry, one time, please, question repeat.
Are there currently any agents in the clinic that can inhibit this exchanger?
Thank you for the question.
What's clinical importance of your research?
Yes, thank you for your question. I think this CGP is very important in a clinical researcher because it is a group of benzodiazepines and may be working in a clinical treatment of acute pancreatitis.
So there is also a question from Alexei Tepikin, who asks, could the exchanger in reverse mode be responsible for the non-MCU mitochondrial calcium entry?
1 second. I read. Okay. Thank you for a question. I think I haven't read anything like it, but I think yes. But we don't have any ideas about it. But it's a good question. Thanks for the question.
Okay.
Thank you very much, Naomi, and we can move and the next presentation. The Masataka Matsumoto presentation effect of recombinant human thrombomodulin on rat experimental severe acute pancreatitis. Please.
Thank you, Chairman. I'm going to talk about efficacy of recombinant human soluble thrombomodulin for severe acute pancreatitis on rat experimental model. Recombinant human soluble thrombomodulin, RTM, has anticoagulant and anti-inflammatory effects and is mainly used for patients with disseminated intravascular coagulation expecting anticoagulant effect. But RTM also inactivates, pumps, dumps, and demonstrates anti-inflammatory effects by activating complements such as HMGB1. Moreover, RTM has reported that ward of necrosis of severe acute pancreatitis, SAP, is decreased by administration of RTM, but efficacy on organ failure has not been demonstrated. The aim of this study is to evaluate whether RTM prevents pancreatic necrosis and remote organ failure of SAP. In this study, we made rats with a SAP model. This is subgroup, and compared the group treated with RTM to the severe pancreatitis model. This is SAP plus RTM group. These manufacturing methods and the schema for this experimental protocol are as shown in figure 2. Results: survival curves. In the subgroup, only 40% of rats were survived 24 hours after the start of the experiment. On the other hand, in the SAP plus RTM group, all rats were survived. Effects on the liver. Macroscopic and microscopic findings, the SAP plus RTM group was able to maintain the normal structure more than subgroup. Serum AST and ALT levels were significantly decreased by the administration of RTM. Effects on the pancreas. Macroscopic and microscopic findings, the SAP plus RTM group was able to maintain the normal structure more than the subgroup, same as effects on the liver. Serum amylase levels were significantly decreased by the administration of RTM. Vascular endothelial cell damage was also significantly prevented in the SAP plus RTM group than the subgroup. Regarding the HMGB1, it was not significant, but mean levels of HMGB1 in the SAP plus RTM group was lower than that in the subgroup. Conclusion: early administration of RTM ameliorates the development of organ dysfunction and pancreatic necrosis, possibly by the maintenance of microcirculation. Thank you.
I have a question. Do you think there is any application that we can imagine for the human disease?
Human?
Yes. Do you think that your finding may be useful to find any application in humans?
In human. As for humans, administration as soon as possible will improve prognosis. RTM can be used to prevent the collapse of microB cells, avoid multiple organ failure at the onset of severe pancreatitis, and reduce mortality. The recommended dose for human and the timing of administration will be considered as a clinical trial in the future.
Thank you. Thank you very much. It's very interesting.
Thank you.
Okay. I think we can move to the next by Sofia Gabriella Pesai on the role of TRPV6 variants in the Hungarian chronic pancreatitis population.
Good morning, everyone. Today I will present our recent findings of the role of TRPV6 variants in the Hungarian chronic pancreatitis population. Mostly genetic variations associated with chronic pancreatitis are located in genes encoding pancreatic digestive enzymes. Chronic pancreatitis genetics is recently seen in a new light since the channelopathy of the pancreas duct is revealed to have a significant act on the development of the disease. The common ground in genes which are related to the ductal pathway is that presumably they have a direct or an indirect impact on the calcium ion concentration in the pancreas duct. Besides the chloride bicarbonate channel CFTR, the cation-selective Claudin 2 channel has been associated with the ductal pathway. According to recent findings, misfunction or decreased expression of the calcium ion channel TRPV6 is strongly associated with early onset non-alcoholic chronic pancreatitis. In our study, we have aimed to verify the role of GP6 variants in the Hungarian chronic pancreatitis population. For that, we have recruited patients from the Hungarian National Pancreas Registry, all patients aged between 18 and 65 years. We will analyze the entire coding region of the gene using PCR and Sanger sequencing in 125 non-alcoholic chronic pancreatitis patients and 230 controls. Based on literature, we have analyzed the exons of GP6 gene with the highest number of functionally impaired variants associated with chronic pancreatitis so far, exon 7, 8, and 11. This table shows the allele frequencies of the mutations we found in the exonic region we have investigated. We did not detect any functionally impaired variants. However, the synonym variant PN504N in exon 11 was significantly associated with the disease. However, further investigation are needed to clarify its role in the development of chronic pancreatitis. Thank you for your attention.
We have a question from the audience. Dora Angiel is asking, why did you exclude alcoholic pancreatitis patients? Could these variants not make these patients more susceptible also to pancreatitis?
Thank you for the question. So the first publication about the GP6 gene regarding chronic pancreatitis was about idiopathic early onset chronic pancreatitis. So we wanted to find out if in the Hungarian cohort, in the idiopathic Hungarian chronic pancreatitis cohort, we can find these impaired variants. And so in Japanese cohort, alcoholic pancreatitis patients were also investigated, but the odds ratio of those patients was very low compared to the idiopathic chronic pancreatitis cohort.
But from the data that we have today, do you think there is a big variability according to the ethnicity, country, or whatever?
Excuse me. So can you just?
Yes, I sorry. According to the data that are already available on this, do you think that there is any geographical difference in the distribution of these mutations?
So we don't know yet. There is a very so we are very lack of data. So that is the one reason we wanted to investigate in the Hungarian cohort, and also we investigate a wider range of age. So between 18 and 65 years. So we just wanted to see if in other patients we could find these impaired variants.
Thank you very much, Sophia.
Thank you.
So we pass to the presentation of Jahedul Alam on the investigation of the endocrine phenotype in the MOD-YH mouse.
Hi, I am Jahedul Alam. So the title of my poster is the investigation of the endocrine phenotype in the MOD-8 mouse. So I am a postdoctoral research fellow from University of Bergen, Norway. So the carboxylic ester lipase or cell gene is the digestive enzyme expressed by the acinar cells of the pancreas. Our research group has shown that mutation in the variable number of tandem region of the cell gene called MERIAD or CELMOD, an inherited syndrome of exocrine pancreas and diabetes. Our previous work has mainly involved in vitro studies. The goal of this project was to develop a mouse model to study the effects of this cell mutation at the organ level. So we created a knock-in mice where the BNTR of the endogenous mouse cell gene was substituted with the mutated BNTR sequence derived from the MODI8 family. We also created a control strain with normal BNTR sequence of human cell gene. If the mice are diabetic or not, we measure fasting blood glucose and perform intraperitoneal glucose intolerance tests. No structural difference were observed between the MODI8 and littermate wild type and control 16R. An increased serum amylase level was observed in the MODI8 mice compared with control, indicating the affection of exocrine pancreas. We further analyze pancreatic tissue. No structural difference of pancreatic islets were observed between the mice strains. In the MODI8 patient, there was decrease in beta cell area and increase in alpha cell fraction compared with controls. So we developed a mouse model for MODI8, which recapitulates the exocrine but not the endocrine phenotype of the disease. So severe exocrine tissue changes or longer observation or different genetic constitution may be needed for this MODI8 mice model to become glucose intolerance. Thanks for listening to me. If you have further things to know, you can see my poster. Thanks.
Shaidul, you only analyzed one patient, or it is a known thing that alpha cells are outnumber beta cells in these patients?
Yeah, it was just 2 or 1 patient, yeah.
But is it known? I'm particularly ignorant in this field, so this is a real question for my ignorance. The involvement of the endocrine system in patients is important. There is diabetes, there is...
Yeah, there was a diabetes. This patient was diabetes. Yeah, that's why we saw that actually diabetes also exocrine change in a patient, but we didn't see the endocrine phenotype change in the mouse.
We also have a question by Yvonne Regel. She asks, what happened when you put the mice on a high fat or high glucose diet?
Yeah, that's it's a nice question. We also tried in a different project. We also analyzed with a high fat diet or high glucose diet, but till now we didn't see any change of endocrine phenotype. That means we didn't see the phenotype of diabetes here.
Okay, thank you very much for the response also. So Sergey, the word to you for the closure of the session. Sergey?
Yes. Okay, we'll listen to some very interesting and promising presentations, and the speakers answered questions to put to them. And so I think we can conclude our section. I wish our participants continued success and all the best.
Thank you. Thank you to all the participants. Have a nice break now. Thank you, Sergey.